Thank you for coming to our presentation. My name is Sarah Nelson. I'm a sophomore biological science major.
My name's Delaney Lantz, and I'm also a sophomore and biological science major.
And our mentor for this research was Dr. Kimberly Hale, and our research is on the effectiveness of various face coverings. So this research actually first began last semester in the fall 22 microbiology course that was taught by Dr. Hale. So all of her students throughout the semester conducted a couple trials, and the first study that was tested focused on the effectiveness of various face coverings, and it ranged from disposable masks, cloth masks, silk masks, different textures, different thicknesses. And so everyone was to follow the same protocol each time they conducted these trials. And in the first study, the student would properly wear their face mask. They would appropriately label a blood or nutrient agar plate using a Sharpie, and then once the plate had been properly labeled, they went ahead and removed the lid, held it approximately 6 inches in front of their face, and coughed hard 3 times. And this was repeated for all the students. And once the plates were completed, they were placed in the incubator at 37.5 degrees Celsius from 24 to 36 hours. And once they were done incubating, they were removed, and colonies were counted if they were present. And we actually found that in this study there was no face covering that was completely effective each time it was used. And then we went ahead and did a second study, and that focused on the amount of microorganisms that were actually stopped by the face mask. And so the way that it was conducted was very similar to the first study, but it had a couple additional steps. So the students again coughed into the face mask and used a plate to collect the data. And once that had been completed, an additional plate that had been appropriately labeled was also used. And so once the student had coughed into their plate, they then removed their mask, and a sterile swab was used to dip into sterile H2O and then swabbed over the inside of their face covering. And then that was then streaked on an additional plate. And those were all placed into an incubator again at 37.5 degrees Celsius for 24 to 36 hours. And once those were completely done incubating, they were removed, and then colonies were counted and analyzed. And we actually found that majority of the face coverings were 100% effective most of the time at preventing microorganisms from getting through the mask. But it is important to note that with the disposable masks, 28.6% of microorganisms were actually able to get through the mask. And it is important that throughout this presentation you keep in mind that the nutrient agar plates can actually only show the bacteria and fungi that are present. It's not going to show viruses. So the only microorganisms that we'll actually physically see on the plates are fungi and bacteria.
So the first study that we decided to do was the effectiveness of various face coverings. So the materials shown here are what were used for the first study. The nutrient agar plates were utilized to see the microbial growth. The sterile swab and sterile water were used to swab the inside of the face covering after it was being coughed through. We used Sharpie to label the appropriate plates and place them in an incubator at 37.5 degrees Celsius for 24 to 36 hours. And then the various face coverings we used were face shield, cloth mask, and disposable masks. When looking at methods, the nutrient agar plates were first labeled with the researcher, the date, and type of face covering being used. We then moved on to holding the nutrient agar plate directly 6 inches in front of our mask, where we then coughed hard for 3 times onto the nutrient agar plate through the face covering being tested at that time. We then moved on to swabbing the inside of the face covering after it was coughed through. We dipped the sterile swab into the sterile water, which we then swabbed the inside of the mask after it was coughed through. We then took the tip of that swab and streaked it across the nutrient agar plate, which we then placed in an incubator lid side down at 37.5 degrees Celsius for around 24 to 36 hours. We then removed the plates from the incubator and analyzed the data, and the data was recorded. So here we have the cloth mask trials. As you can see, the cloth mask cough actually had 1 colony form, which was very uncommon in our data and uncommon in our research. It was actually an outlier during the entire process. But as you can see here, we had we had one colony form that was 5 millimeters. And then, as you can see, our cloth mask swab actually had several different colonies form, ranging from 61 colonies at 1 millimeter to 4 smaller colonies that were less than 0.1 millimeters. And that was the swab of the inside of the cloth mask. And many would assume that during this, the researcher was not expelling the proper microorganisms in order to test this. However, the colonies formed from the swabbing of the inside of the cloth mask do show that the researcher was expelling the proper microorganisms. However, the mask was just effective in stopping those organisms. Here's some representative plates. On the left, that is our outlier in our data. That's the one colony formed through the coughing of the cloth mask. And so that is very uncommon, but as you can see, we do have a bigger colony formed on that plate. But as you can see the difference in the right plate where we have several colonies formed in the swabbing of the inside of the cloth mask. So moving on to the data of the disposable mask trial, we can see that through the cough of the disposable mask, we had 0 colonies formed, which does show the effectiveness of disposable masks in general. And as you can also see, the swabbing of the inside of the disposable mask does show several colonies, also showing that the researcher expelled proper microorganisms throughout this research. And you can see that we had a big range. We had 15 colonies at 1 millimeter with smaller colonies surrounding that. And then we also had smaller sporadic colonies that actually were too small to be measured on that plate. And then moving on to the representative plates, on the left you can see that we had 0 colonies form. And then on the right is the different sporadic colonies that we had from the swabbing of the inside of the disposable mask. Finally, moving on to the face shield trials, we can see that the face shield was effective in stopping the microorganisms through the cough, as it did have 0 colonies formed on the nutrient agar plate. But as you can also see, we had colonies formed from the swabbing of the inside of the face shield. We did have, however, less colonies formed from the swabbing of the face shield compared to the cloth and disposable mask. Overall, the colonies, we did have fewer colonies, but they were still present, showing that microorganisms were still expelled by the researcher. So as you can see on the left, it's harder to see because we have smaller colonies, but the swabbing of the face shield does have colonies present on the left. And on the right, you can see that we did have 0 colonies formed from coughing through the face shield onto the nutrient agar plate. So then we decided that it would be significant to add in this study what actually happens when you double up the different face coverings. So the materials here were the same as study 1, however we did use double the face coverings in order to test that theory. When looking at the methods, they were very similar to study 1 as well. However, when coughing onto the nutrient agar plates, we were either doubled up with 2 disposable masks, or we had a cloth mask and a face shield on. And then after that, we moved on to holding the nutrient agar plate directly in front of us at 6 inches, which we then coughed onto the nutrient agar plate. We then moved on to swabbing the inside of the mask, and we also thought it was essential in this part of our study that we actually swabbed the inside of the inner disposable mask and the inside of the outer disposable mask when using the disposable mask, and the inner of the cloth mask and the inner of the face shield when using that part of the study. So when doing the methods, as we took the sterile swab, dipped it in the sterile water, swabbed the inside of the inner mask, the inside of the outer mask, and streaked them onto their appropriately labeled plates, which we then placed in an incubator at 37.5 degrees Celsius for 24 to 36 hours, lid side down. We then pulled them out and analyzed the data, and the data was recorded from that. Looking at the double disposable mask, we can see that coughing through the 2 disposable masks did give us 0 colonies on the nutrient agar plate, which we did expect considering that we also had 0 growth from just 1 disposable mask. So we also assumed that we would get 0 colonies from the double disposable mask. We can also see that from the swabbing of the outside mask that we did have 0 colonies form as well, which is very significant because this shows the effectiveness of the disposable mask because the microorganisms were not even able to get through the mask onto the second mask. And as you can see here, the proper microorganisms were expelled during this part because we did have colonies form on the inner disposable mask. So therefore, it was effective in stopping the microorganisms from expelling into the air via respiratory droplets. So here are some representative plates of that. On the top right, we have the double disposable mask, cough, which we had, or on the left we had the cough, which was 0 colonies. And then in the middle, we had the swabbing of the outside disposable mask. And then the top right were our colonies formed, which was the swabbing of the inside of the disposable mask. So next we moved on to the face shield and the cloth mask, where we then did the very similar thing, but with face shield and cloth mask. As you can see, the coughing through the cloth mask and the face shield did form 0 colonies. And the face shield swab also formed 0 colonies, therefore the same thing that the cloth mask was effective in containing the microorganisms from getting through the cloth mask onto the face shield. And as you can see, the microorganisms were expelled by the researcher due to the fact that colonies were formed from the swabbing of the cloth mask. And as you can see, we had 10 1-millimeter colonies from the swabbing of the cloth mask. So some representative plates. In the middle, we can see the colonies that formed from the swabbing of the cloth mask. And on the right and on the left, we have the 0 colonies formed from both the coughing through the cloth mask and the face shield and the swabbing of the face shield.
So then we went ahead and did a third study, and we wanted to focus on what would happen if we were to test this over various distances. So in this illustration, it shows what actually happens when someone coughs and expels respiratory droplets. So you can see this person on the left is coughing, and these are the respiratory droplets that are being expelled. And it also demonstrates the 3 common ways of transmission of SARS-CoV-2, which here we have the short range, here we have on the surfaces, and here is the long range. And as you see, it is 6 feet away from the person who is expelling the respiratory droplets, which is the recommended social distance of the CDC. So the materials for this study were the same as study 1 and study 2. However, we only used a disposable mask. We did not use a face shield or a cloth mask in this study. And we also used 2-meter sticks in order to measure the distances that we wanted to test. And the methods in this study are also very similar. There are a couple differences, however. So again, we appropriately labeled our nutrient agar plates, and then once we were outside, we placed the 2-meter sticks on the ground so that we were able to measure the distances that we wanted to test. And one researcher who was coughing either placed the appropriate mask on or completely removed their mask depending on which trial we were conducting. And the other researcher would hold the nutrient agar plate, remove the lid, and hold it in front of the other researcher who would then go ahead and cough at the appropriate distance. And then once all of the 7 nutrient agar plates were completed, we brought them inside, placed them in the incubator at 37.5 degrees Celsius for 24 to 36 hours. And then once they were completed in the incubator, they were removed and colonies were counted if they were present. And so the first trial that we conducted, we used no mask, and surprisingly, we actually found that in all 7 of the agar plates, there were 0 colonies, and we tested the closest at 6 inches and the farthest at 6 feet, which is the recommended social distance. And it is very important to note that when we actually conducted these trials, there was a significant amount of wind, and it is possible that that could have skewed our data because the wind may have moved the respiratory droplets that were being expelled. So here are some of our representative plates. As you can see on this left side, this is the closest distance that we tested, and there are 0 colonies formed. And on the right one, this is the farthest distance that we tested, and again, there are 0 colonies formed. So then we went ahead and we did the same thing. However, this time we used a disposable mask. And because in the previous trial, without a mask, we saw zero colony growth, we assumed and hoped that we would also see the same thing when using a mask. And that's exactly what we saw. For all 7 of our nutrient agar plates, they had zero colony growth from the shortest distance all the way through the farthest distance. And again, it is important to note that we did have a significant amount of wind when we conducted this trial, which could have skewed our data. And so here again, we have some representative plates. On the left side, we have the closest distance that was tested, and on the right side, we have the farthest distance that was tested, and both of those show zero colony growth. And so then finally, we wanted to use a dirty disposable mask, so we did the same thing. However, this time, the researcher who was coughing through the mask used the same disposable mask that they had been using all throughout the day and up until the time that we were testing. And we again saw that there was zero colony growth on all 7 of the nutrient agar plates. And just as the other 2 trials had issues with wind, we also had a significant amount of wind during this trial, so it is possible that this also skewed our data. So here we can see our representative plates. On the left side is the closest distance tested, and on the right side is the farthest distance tested, and in both of those there is zero colony growth. And because on our initial trial we didn't have any microbial growth from testing without a mask, we felt that it was important that we conducted this trial a few more times just to see if we could actually see some microbial growth. So we had a total of 4 nutrient agar plates that were tested at 6 inches, and in one of those, which you can see here, we did have some microbial growth. There are some colonies throughout. So it is significant that we tested this just to show that there are actually microorganisms in the respiratory droplets that were being expelled when we conducted these trials. So we also think that it's important that we tested some disposable masks directly from an open box of disposable masks, because so many businesses do leave open masks out in front of their entrances solely because a lot of people tend to leave their personal masks at home when they come into places of business. So we did see that there was some microbial growth from one of the masks that we tested. We can see this colony formed right here. So that is very significant because it does show that in an open box of disposable masks that may come into contact with other people, that those masks are not necessarily sterile and they could have microbial growth on them. We all have a natural flora that's on the surface of our bodies, including our hands. So when people come and grab masks and potentially just touch near the box, there is the possibility that microorganisms from their natural flora do get onto the masks. And we also understand that some people do have concerns with the microorganisms that actually grow on the inside of your mask. And just as I said in the previous slide, we do have a natural flora inside of our body as well as on the outside. So when we actually cough and expel respiratory droplets, the microorganisms that are present in those respiratory droplets are our natural flora and are inside of our own respiratory system. So the microorganisms that are going to be growing on the inside of your mask when you wear them all day and you cough into them are actually part of your natural flora, and they do not pose any additional threat to your health.
We also thought it would be significant to add in our study oxygen particles versus viruses. If oxygen particles are unable to get through a mask, how could viruses? As stated previously in our presentation, that on our growth of our plates, we could only see the growth of bacteria and fungi because we could not see the growth of viruses. In this video, we will be shown one trying to blow out the flame of a candle while wearing various face coverings, but he is unable to do so because oxygen particles are unable to travel through the mask. Oxygen particles are much smaller than viruses due to the fact that viruses often have to travel on molecules. Viruses are much smaller than some microbes. However, viruses are still large enough to not be able to get through a mask, and this will be shown in the video. We also conducted this with 11 researchers in our group. that had various face coverings on, and each went through and tried very strenuously to try to blow out the flame of the candle and were unable to do so.
So in our summary of findings, we did find that various face coverings are effective at stopping the transmission of microorganisms through respiratory droplets, and we also found that doubling up on face masks is also effective in stopping the transmission of microorganisms through respiratory droplets, but we also did not see any increase in the effectiveness when using 2 face coverings at the same time.
When looking at our future studies, from what we found, we thought it would be interesting in order to test from the sides of the mask, considering that we only tested approximately 6 inches in front of our face in front of the mask. And we are unaware of the microbial growth that comes from the side of the mask, so we thought this would be very interesting to further our research on the overall effectiveness of various face coverings. So moving on to acknowledgements, we would like to acknowledge the microbiology class from the fall of 2020. They actually started the research for us and gave us more ideas on how to further the research. We'd also like to thank Dr. Hale's research group in general, because when we were unable to be there, they did a small variety of things for us. Thank you. So in general, we think it would be beneficial to do it inside, but with the social distancing requirements right now, we were unable to do it inside. So we thought our best bet would be to try it outside, even though it was a little bit more inaccurate.
Policy on face masks, yes or no?
Yes, so we did actually see that it is effective when you do use a face mask, because there was a significant amount of microbial growth that was stopped when we tested using face masks. So it is effective. One possible thing is because of, it is a different, like we had cloth and disposable, and that is more of like a solid type of surface, but it's also possible that the way it's structured, there might be the possibility that microorganisms possibly spread out through the sides more so than they did in the other masks. So that's also something that we would potentially find in our further studies.
Also, to add to that, I just want to emphasize that a face shield is, if you think of a face shield, it's about right here on your face, where a mask in general is confined across your whole face. So I think that the microorganisms, when looking at the mask, go directly into the mask, whereas the face shield has more openings, as Sarah said.